Quick update on who Minoryx are. We're a very small Spanish company, founded about just 10 years ago, just outside Barcelona. And we have one particular product in development, which is liraglutide. And we have activities with the EMA and FDA orphan designation in both regions. So we've got pathways to get a product to approval, which is our aim. And then liraglutide. It does have a future trade name if we get an approval. So it will be called Nesglial. It was named before I arrived, so there must be a reason why it's named that. It's actually a metabolite, which it means it's a product of another drug which is already used in clinical practice for diabetes. So its parent drug is called pioglitazone, and liraglutide is a metabolite of pioglitazone. So when the body is metabolizing pioglitazone, one of the byproducts is liraglutide, and that's a product that we've selected to take forwards into development. Why liraglutide? Well, we've looked at it in terms of how it penetrates the brain. It penetrates the blood-brain barrier and has higher levels in the blood-brain barrier than pioglitazone does. Also has a multi-mode of action, which I'll take you through shortly, again in the diagram, but it reduces inflammation, improves myelination, reduces demyelination. So it's multi-action type of product, which is quite interesting. At the moment, we're in a sort of interesting time point with the EMA. We have actually submitted our dossier for an approval to EMA, and we're in dialogue with them, answering questions. If everything works out, there may be an approval for the product in, we'll have to see what the EMA say, but within a subset of ALD patients, maybe by the end of the year or early next year. So we're keeping our fingers crossed, and I'll take you through the data that is being reviewed by the EMA. The UK licensing will follow that because the EMA activity comes first. The UK MHRA will look at the output from the EMA, and it's a shortened review process, but it's still, unfortunately, sequential as opposed to in parallel. And then also we're working with the FDA. I'll take you through a new study that's going to be set up for the US in the near future, starting later this month, early and in the next couple of months. Okay, and then also importantly, I think you've seen that we have a partner company here in Europe called Minoryx Pharm. Colleagues from Minoryx Pharm have been here, and they will be the company that will be launching the product across Europe if we get the approval. Okay. So this is a complicated diagram, and really just to say, this is a cell. The darker pink section in the middle is the nucleus, and liraglutide acts through the nucleus, adapting the outputs from the nucleus in terms of messengers, reducing the inflammation, acting on the mitochondria, which are the energy-generating sort of organelles within the cell, and also sort of changing things which within the CNS the cell can do in terms of reducing the stress, as I've said, reducing the inflammation, and really sort of adapting the cell to survive a bit better. And hopefully that's what we do in the CNS. We have preclinical mouse model data and cell data which supports this activity. And now we have some of the clinical trials which I'll take you through in a second. I'm not going to educate you on ALD and AMN. I think you've had lots of presentations, but really just to show you that within CALD, within cerebral ALD, the activities that we see from our preclinical models are around the endothelium, which is the the lining of the blood vessels, which protects and acts as a blood-brain barrier. There's some activity there. Then also activity in the microglia to stop the inflammatory process, and stabilization of the oligodendrocytes, and a slowdown of the demyelination, and then possibly some remyelination as well. And then also in AMN, so I think we're pretty unique that we have a product that we think works across the whole of ALD. Not on very long-chain fatty acids, so that's the key thing. It's a central nervous system activity. In AMN, we're looking at the spinal cord here. I think you heard the presentation from Dr. Eichler. It's still part of the CNS. I think one of the things which is really important for AMN is that the natural history for AMN isn't well understood. And that will sort of become apparent as I take you through the data. So the two things that I'll be talking about are the ADVANCE study, which I think has been presented here before. And this is a study in adult AMN patients, men, which was run across Europe and the US. Actually with 116 patients, so the largest ALD study thus far that we've seen published. And then also a study in pediatrics, which is ongoing at the moment, called Nexus. The AMN study, the ADVANCE study, has been published, and all the data I'll be presenting is already in a paper. The Nexus data that you'll see was presented just last week on Tuesday at the American Academy of Neurology. Okay, so the ADVANCE study, which was a study set up to look at progression of adrenal myeloneuropathy and the effects of liraglutide. And it was a placebo-controlled study, so as you can see, with a 2-to-1 randomization, more patients on liraglutide, twice as many as on liraglutide compared to placebo. And then there was a 2-year period of treatment, followed by an open label extension, which meant all of the patients who were on placebo could take the active drug going forwards. We were looking at a primary endpoint of the 6-minute walking test, which I think you've probably heard about through the course of other meetings. But it's a typical test that the regulatory authorities, the EMA, FDA, use for neuromuscular diseases, genetic diseases like Duchenne's muscular dystrophy. But it isn't applicable to all disease states. And I think when you've got a progressive disease, you always catch patients who are at different stages of the disease, which becomes apparent as we take you through the data. The other endpoints that we were looking at were body sway, which is really a measure of balance. And then some of the typical clinical scales like SS-PROM, EDSS that are used in neuromyopathies. And then some of the clinical physician input in terms of global impression and the patient feedback from quality of life and some other sort of quality of life measures. Also, importantly, we were measuring any cerebral lesions that were appearing or present and seeing if they were changing in any way. So we were doing regular MRI scans in these AMN patients who were not diagnosed with cerebral ALD. This is really just to say we screened 136 patients, we randomized 116 patients, 77 patients received liraglutide, and 39 patients received placebo. I'll keep going. This is really just to say the patient groups between liraglutide and placebo were comparable, so there was no sort of oddness in how they were randomized. One important thing that you can sort of see right down here at the bottom is the less score. What we found when we did the MRIs at baseline, this was before anybody had any treatment, was that more than 50% of the patients had a less score of greater than 0.5. So there was something already apparent in the MRI scans. There were no signs or symptoms of cerebral lesions, cerebral disease as such, but we saw this on the MRI scans. So the results. So, unfortunately, as you can see from this top graphic, there was no difference between liraglutide and placebo on the 6-minute walk test. The outcome was there was no difference after 2 years of treatment, which is a little bit frustrating after 2 years of running a study, knowing that. But whilst the study was being set up, there was, as I said, very little in the way of AMN natural history. And during the course of the study, natural history started to become more apparent, particularly from the Dutch group, where there was some work ongoing, that the disease in AMN is not a linear disease. There is much more disease that happens early on from the time of initial symptoms, and then that sort of stabilizes, especially as patients become less ambulatory. So we started looking at patients who had less than 10 years of symptoms, and when we start doing that, as you can see from this graphic, that there starts to be a trend. It's not a significant difference, but a trend that there is something happening even on the 6-minute walk test. So we can't claim it's a success with that. So the study didn't meet its primary endpoint, which is always, for the regulators and scientifically, well, you didn't set the study up properly, or you weren't looking at the right things. In a rare disease, often you're not always looking at the right things because the disease needs to be better understood. And I think you've heard from a lot of the experts over the last 2 days that natural history studies running in parallel to development programs is now the norm. When we started this study 5 years ago, it wasn't really the norm, unfortunately. So I think it's a lesson from this study that doing that in time, on time, is important. So when we look at the other outcome measures, the secondary outcome measures, in terms of body sway, which when we look at what patients say with AMN, balance is a primary problem. It's an early problem, and that remains as the disease progresses. More walking aids are needed. Patients fall. We see fractures. And I think just 10 days ago, ALD Connect published the patient-focused drug development discussion and presentation, and that was really sort of, please do take a look at that. I think Kathleen hasn't spoken about that, but I think if you take a look at the ALD website, ALD Connect website, there's 90 pages of really valuable information there. And we're using that to support our dialogue with the EMA and FDA. But when you look at body sway, which is assessing balance using what we call a force plate, and the force plate is really just a static plate. You stand on it, and then if you close your eyes, put your feet together, you will sway, and it assesses where are you putting your pressure points and how much are you moving about. So it's sort of assessing, as you can see from this little graphic on the bottom left, it just assesses forwards and backwards and sideways movements. And really the critical one that is probably the most sensitive is eyes closed, feet together. So if you think about closing your eyes, putting your feet together, and just trying to stand still, if you have got balance problems, that will be picked up. And this is quite an objective measure compared to just saying, how is your balance to a patient? So I think it's something that we can actually measure very, very clearly. When we look at body sway with especially the eyes closed, feet together, which is the bottom row, we actually see an improvement, which is sort of slightly unexpected, in that we see an improvement in a disease that is progressive and typically irreversible. So we need to sort of understand that, but it may be just that different muscles are being improved, or we're not quite sure why there's an improvement. But for us, even stopping the worsening is really important. I think that's quite key. So body sway, balance, we think is a really key symptom and endpoint to look at. And then when we look at the typical endpoints like EDSS, which is typically a clinical scale used in MS, and I think a lot of the doctors will be using it within AMN patients as well, and also quality of life. And we look at the top line is all of the patients, and then the bottom row of figures and graphs is where the patients just with less than 10 years of disease are presented. When you've got less duration of disease, you start seeing a significant difference, which is really important and interesting to sort of start understanding how the disease sort of isn't a linear disease, especially for ambulation. So I think these are important points that we're understanding better. And then when we sort of put all of the pieces together, AMN is multifactorial. It doesn't have one key sign or symptom. It's progressive. So looking at all the different variables, we get a trend. We don't get significance, which is a key scientific and regulatory parameter, but we get a trend, which is quite good for SS-PROM, another clinical scale, which is quite intensive in terms of collecting the information, EDSS, which we've talked about, and then quality of life as well. There's a trend after 2 years of less worsening in quality of life. So we have to look at the whole picture for the data. And then moving on to that final piece I said about the cerebral lesions. So the patients came into the study and no patients had cerebral ALD. So no patients had the progressive form of the disease. But what we found was patients did have lesions that at baseline were noted as being not active. And what we saw, first of all, the graphic on the left, is the incidence of cerebral lesion progression. So that means the less score progressing. Yeah. And what we saw was 3 out of 77 patients in the active group compared to 8 out of 39 patients in the placebo group. So we saw more in the placebo group progressing in terms of the lesions progressing with the less score. And you can see that in the middle graphic as well. In addition, what the investigators were collecting was the clinical diagnosis of cerebral ALD. So really just assessing that there was something much more going on than just a brain image in terms of the clinical signs and symptoms of progressive ALD, and also together with the MRI scans where there was starting to be gadolinium positive enhancement. And what we saw there was there were no patients in the leraglitazone group, but 6 patients in the placebo group developed CALD, which is clinically and on imaging confirmed. Okay, after 2 years. So that was, again, unexpected, but more than interesting in terms of reducing the incidence of the cerebral disease in AMN patients. Some of the other things that I think you've heard about over the last 2 days is that there are other biomarkers, neurofilament light, and looking also at the blood-brain barrier. There is a biomarker called MMP9, and we saw differences between the treatment group and the active group there as well, and the placebo group there as well. Of course, we do know that there were those 6 patients who were having more in the way of cerebral progression. They're captured in here as well, so they do give that difference, but it's just saying that there are other biomarkers that are important, as well as MRI scans. Okay, then onto the safety side. So safety-wise, it's an oral liquid, so it's not an injection, it's not into the spine. This is once a day as an oral liquid. It's a drug that the class is known, so PPAR gamma agonists within the diabetic field, and we know that they cause edema and weight gain. They caused edema, the product caused edema and weight gain. The edema and weight gain didn't sort of cause bigger clinical problems. So we were looking out for heart risk increasing because of fluid retention, which is what edema is. That wasn't a problem. There was no kidney problems with that either. And with weight gain, yes, it becomes annoying, but it's something that, again, happened quite frequently. Otherwise, the product actually behaved quite well. There were no cardiovascular issues, no liver issues that we found with ongoing treatment. And the other thing that we were doing is looking at the urine for cells which were possibly changing, because this class of drugs is also known to have an effect on the bladder and possibly bladder cancer. So we've been monitoring that quite closely, but there have been no cases thus far. So overall, edema and weight gain were the key ones. Some increased tearing, so lacrimation, so more tears, which was a little bit annoying for the patients. The edema was annoying, bothersome. Some patients did discontinue from the study because of that. And the weight gain actually stabilized after 2 years. It's because PPAR gamma agonists cause an increase in fat tissue. So as well as the fluid retention, they can cause fat tissue accumulation as well. Okay. So now I'll move on to the Nexus study. And this is a study that's ongoing. It's an open-label, single-arm study looking at liraglutide in the treatment of childhood cerebral adrenoleukodystrophy. And as I said, this was presented by Dr. Malak, who's a pediatrician from Cornell in New York and has been working on the study. I won't go through this except this figure on the bottom right, which I think you had an explanation from Professor Kohler yesterday as to what these Kaplan-Meier curves look like. And this is really just data showing differences between overall survival and major functional disabilities in untreated pediatric patients. So there's a decline in the lines, as you see, because without treatment, patients do unfortunately die, and they also have significant morbidities in terms of disabilities. Okay, so the study is quite simple. There's a screening period, and then patients are treated once a day with liraglutide. We were looking at patients who had either just cerebral lesions identified, but no gadolinium enhancements and not the inflammatory effect. So these patients obviously are being screened, or they're coming in with an MRI scan, either that's showing inflammation or not showing inflammation. So we've got two groups. And what we wanted to also look at is to make sure that the patients didn't have any neurological deficits, so they were coming in quite well, and because that's one of the endpoints that we want to measure. We also want to look at safety because of what we've been seeing through the advanced study, and also to see in terms of the actual endpoints, can we slow or arrest the disease progression, both on imaging and also clinically. And what I'm about to present is the first 24 weeks of data, which was set up really looking at safety and also should we continue the study. Because if we were not seeing some level of effects, then we shouldn't continue and expose patients unnecessarily to something that may not help. So we wanted to see if the study continued, if more than 40% of the patients showed any level of lesion deceleration or arrest of disease. So far, we've actually recruited 20 patients up to this month, and the study is about to close. The study's been recruiting in France, Spain, Germany, and also in Argentina. Okay? So, first of all, on the safety front, we've not had any withdrawals due to adverse events. The pediatric safety profile is less of a concern to us than the adult profile. We've only had 3 cases of edema and 1 case of weight gain. So on the growth charts, the boys are following their typical growth as opposed to shooting up when they've been exposed to liraglutide. There's been no cardiovascular problems or hepatic problems, so we think at the moment it's tolerated quite well. What you can see on the bottom left, and as I've said, presenting 24-week data which is there, but we actually have some patients who've gone further than 24 weeks, and that first graphic is about the less score, and you can see that typically the less score does increase a little bit and then stabilizes. And this is all of the patients, both gadolinium enhancing and not enhancing. So we're quite comfortable with how that is looking. It looks similar to what happens after bone marrow transplant, after HSCT. So at the moment it's following that pathway, which is reassuring. And what we actually sort of stated as part of the outputs is the continuation criteria are met, so the study is continuing. All patients are clinically stable, so they're not showing signs of any neurological deficit. There have been no major functional deficits identified. The lesions are actually slowing down in terms of their growth rate, so that's important because we're actually looking at lesions that are not just through the less score, but volume, which may become the future gold standard, because the less score doesn't look at how big the lesion is. It looks much more at where they are, the number of different areas affected. So looking at the volume of lesions is going to become more important, and I'll share that with you in a second. Just now. So what you see on the bottom left is a graphic. You see the dotted lines across the middle, and that's the natural history of how the lesions are expected to grow, which is a 2 or 3 fold change on per month. So that's the monthly fold change, so it's like the volume change. So what we see with liraglutide is there's a deceleration of growth, and the majority of patients are maintaining that deceleration and slowing of growth towards 0, which is, again, reassuring. We do have this one patient that has taken what we think is the natural history and has not responded, seemed to respond initially, but has taken off after 6 months. And we're sort of following that case carefully. Also, when we look at the neurofilament light chain, and this is that biomarker, again, very reassuring. Most of the patients are maintaining their initial levels. They're not worsening. So again, that's another key signal to say there's an ongoing effect. So really, just to conclude, the study's carrying on. We're really pleased that the tolerability, the safety profile, is a little bit better than it is in adults, which is good. But we'll carry on monitoring. We'll expect the final results in 2025 when this last patient from this year finishes 2 years of treatment. Last 2 slides, very quickly. We're setting up an adult CALD study, so adult cerebral ALD study in the US and Latin America, which will start this month, next month. This is a placebo-controlled study, and the primary outcome is survival. So it's very much like an oncology study, and we've had to do this in terms of how we've had feedback from the FDA with the lack of natural history data. Very little is understood as to how adult CALD progresses. So we're looking at this very prospectively, but the study has been designed to have an interim analysis at 18 months. And if that is successful, we actually change over that all the placebo patients will start receiving active drug. If that is not successful, that can happen again at 27 months. They'll flip over. If that's not successful, the study will run its full course to 36 months. We'll be looking for 40 patients in total, 20 patients on active, 20 patients on placebo. And the study is going to be running, as I've said, in North America. So at Stanford, in Utah, Minnesota with Troy, at Florian Center in Boston, at Kennedy Krieger with Ali Fatemi, also in Florida, and then 2 sites in Brazil and Argentina. With that, I'd like to, first of all, thank everybody for listening, thank all the patients and families and investigators who have been working on quite a critical program and a lot of intensive work for the past, I'm trying to think, 5-6 years in collecting all of this data. So thank you all very much.